Welcome to Wastewater 101, a series of videos designed for health department professionals engaged in wastewater surveillance work and interested in learning more about wastewater and what their utility partners do. This is part 5, How is water quality monitored? I'm Anna Marotra, Director of the Wastewater Surveillance Program at the Water Environment Federation, or WEF. And I should note that development and production of this material was made possible through funding from the US CDC to WEF under Cooperative Agreement CK 2020-03, but this material is solely the responsibility of WEF and does not necessarily represent the official position of CDC. So just like for wastewater surveillance samples, water quality monitoring samples in the wastewater collection and treatment system can be taken from different matrices. Usually these samples are either taken from a liquid matrix or a sludge or solids matrix. So liquid samples can include things like raw influent, primary effluent, mixed liquor suspended solids or MLSS, secondary effluent or final effluent. And sludge or solid samples can include things like septage, primary sludge, waste activated or return activated sludge, and thickened sludge. And the solids content can vary from less than 0.01% for secondary effluent and final effluent to about 0.01 to 0.03% for raw influent and higher for MLSS. And just a note that 1% solids translates into 10,000 milligrams per liter of total suspended solids, or TSS. And we're going to talk about TSS in just a minute. And just a reminder that we cover the different wastewater unit treatment processes back in part 3 of this series. So that's where we talk about things like primary clarifiers, primary effluent, secondary effluent, and things like that. And I should note that sludge concentrations can certainly be higher than 8% once the sludge is put through a dewatering process. But usually once it's dewatered, the sludge is no longer called sludge, it's actually called cake or dewatered solids. And in that case, the solids content can get as high as 15 or 20%. Also, just like for wastewater surveillance samples, either grab or composite samples can be collected to monitor the quality of water through the wastewater treatment process. Grab samples are usually collected for sludge and also for liquid samples that are going to be analyzed for parameters that aren't stable over time, such as temperature, pH, or nitrate. Although grab samples can sometimes be used for raw influent, composite samples are more common. Most composite samples are probably time-based composite samples taken over a 24-hour period, which means that a certain volume of wastewater, maybe 100 mils or so, is collected every 15 minutes or so. But many plants, especially the larger ones, take flow-paced composite samples. So for flow-paced composites, a fixed volume of sample is collected every time a predetermined volume of flow has passed the sample point. So that obviously requires the use of a flow meter and feedback from that flow meter to the composite sampler. So here are some photos of grab sample techniques. Composite samples usually involve the use of automatic samplers or auto samplers. At the influent to a treatment facility, the auto sampler is likely a refrigerated unit set up in a doghouse or shed, like the first 4 images shown here. Also, the auto sampler is usually configured to collect samples into a single 10-liter carboy, although they can also be configured to collect samples into 2.5-liter carboys or 24 discrete samples, like what's shown on the right. So 1 sample per hour. There's a lot of flexibility in how an auto sampler can be used. So when it comes to analytical methods for monitoring water quality, there are 2 places that wastewater and environmental labs go for these methods. The first is EPA's Clean Water Act analytical methods, and the second is standard methods. So related to the first, EPA approves methods that can be used to evaluate specific analytes in water. Usually the US EPA approves 1 method per analyte, whereas standard methods for a given analyte may have multiple methods listed, including the EPA approved method. Because standard methods then has more options per analyte, and it really is the most commonly used analytical methods reference in wastewater and environmental labs, I'm going to talk mostly about standard methods. But you can find the EPA approved methods at epa.gov/CWA methods, and you can find standard methods at standardmethods.org. It does require a subscription to access all of the methods on standard methods, but you can read the introductions to each method for each analyte without a subscription. The latest version of standard methods, by the way, is the 24th edition, published in 2023. And it is a joint publication by the American Public Health Association, the American Water Works Association, and us, the Water Environment Federation. Standard methods has many chapters, with each chapter covering a class of methods as listed here. Now, I'm not going to talk about all the methods, but I'm going to provide a little bit more information on 4 classes of methods, namely physical and aggregate properties, inorganic nonmetallic constituents, aggregate organic constituents, and microbiological examination. If we look at the physical and aggregate properties chapter, we see methods for all of the properties listed here, from appearance to dissolved gas supersaturation. And again, I don't want to talk about all of these, but I do think it's worth spending a minute or two on solids. And the solids measurement that you probably hear most often is total suspended solids, or TSS. And I've already mentioned that earlier. TSS combined with total dissolved solids, or TDS, equals total solids. That makes sense. But when it comes to influent wastewater, what we usually care most about is TSS. And in fact, that's what we usually care most about all across the treatment train. This diagram shows how TSS is measured. Basically, a known quantity of wastewater sample is passed through a prepared and weighed glass fiber filter paper with the help of a vacuum pump. After filtering, the filter paper, along with the solids retained on that filter, are dried at 104 degrees C until all of the water is evaporated. And the solids remaining after evaporation are considered TSS. The solids that pass through the filter into the flask are TDS. Again, that's total dissolved solids. So you can see that the cutoff between suspended and dissolved solids depends on the effective pore size of the filter used. Now, standard methods list examples of suitable glass fiber filters for TSS measurement, but the pore sizes of those range from 1 up to 1.5 micrometers or microns, and sometimes even higher. It just depends on the manufacturer of the glass fiber filter. So it's possible and even likely that colloids, bacteria, and other things that might be considered more a solid will actually end up being counted as TDS. And just to be clear, in an influent wastewater sample, most of the time, neither the TDS nor the total solids, so that would be the amount of solids remaining after evaporation of an unfiltered sample, is quantified. TDS might be approximated by measuring electrical conductivity, because the higher the TDS content, the higher the conductivity of a sample. And although electrical conductivity measurements may give some indication of the potential for PCR inhibition, which increases with increasing salinity, even electrical conductivity measurements are not uniformly performed for influent wastewater at all treatment plants. And I've noted here that the typical influent TSS values, again, this is for raw influent, are in the range of 100 to 400 milligrams per liter. They can be even lower than that during precipitation events in a combined system. And we talked about combined versus separate collection systems back in part 2. So why spend so much time talking about TSS? Well, solids are really important for the fate and transport of pathogen markers in wastewater. So if we're talking about viruses, then the research suggests that virus particles, whether coming from enveloped or non-enveloped viruses, appear to preferentially partition to solids in raw wastewater rather than hang out in the water itself, just the liquid part of the raw influent. Mercier et al., for example, showed that for one treatment plant in Ontario, Canada, more than 80% of the endogenous influenza A viral signal, that is the influenza A present in the raw wastewater and not spiked into the wastewater sample, was partitioned to the solids fraction. And Ralden, Hernandez, and Baim showed that for 6 California treatment plants, endogenous RNA concentrations of SARS-CoV-2, RSV, rhinovirus, and F plus coliphage were 1,000 to 10,000 times higher in the solids fraction than in the liquid fraction. And if we're talking about bacterial pathogens, whole bacterial cells may be right on the cusp of being captured in TSS measurements or not. But certainly bacterial flocks or agglomerations of bacterial cells that are loosely associated with each other will be counted as TSS. So it's just really helpful to know how TSS is measured, as well as what the TSS values are for your wastewater surveillance samples. So going in order of the chapters and standard methods, the next category of analytes worth mentioning is the inorganic non-metallic constituents, specifically nitrogen and phosphorus. When it comes to nitrogen in raw influent wastewater, most of it is what we call total Kjeldahl nitrogen, or TKN. TKN is the sum of nitrogen bound in organic substances and the nitrogen present as ammonium, that's NH4 plus, or ammonia, NH3. And just to note that because the pH of most environmental waters, including wastewater, is close to 7, most of the ammonia species in wastewater is actually the ammonium ion, or NH4 plus. TKN measurements are performed by adding sulfuric acid to the sample and heating it to digest organic matter and liberate the ammonium. The ammonium is collected and measured, which indicates how much nitrogen you originally had in your sample. And this ammonium can come from different types of organic nitrogen in your wastewater sample, including things like urea, which is CH4N2O, amino acids, proteins made out of those amino acids, and even ADP and ATP. TKN is usually 60 to 70 milligrams per liter in raw wastewater. That's milligrams as nitrogen. And about 60 to 70% of that TKN is not organic, so just ammonium. I am showing here a little sliver of nitrate and nitrite, that's NO3 minus and NO2 minus, but in reality, there's usually no nitrate nitrite in raw influent because the sewer is a reducing environment. And so nitrate is an electron acceptor. It can be used for respiration by many bacteria, but because of the conditions in the sewer, it's usually been used up by the time it gets to the treatment plant. Just like nitrogen, phosphorus has different species in wastewater and in water in general. In raw influent, if phosphorus is being measured, then it's most likely being quantified as total phosphorus, or TP. So that includes phosphorus bound in organic matter, but also orthophosphate, so that's versions of PO4 3-, and polyphosphates. Those are orthophosphates that are just strung together in a longer molecule. TP is usually about 4 to 12 milligrams per liter in raw influent, and that milligrams of phosphorus per liter. And about half of that is usually the dissolved orthophosphate species. So moving on to the next class of methods, I wanted to talk about the aggregate organic constituents, most notably biochemical oxygen demand, or BOD, and chemical oxygen demand, or COD. Both of these measurements are getting at the same thing, the amount of biodegradable organic matter in the sample expressed as oxygen demand. So it's worth talking about what exactly oxygen demand is before we get into more details about the BOD and COD tests. So we're all pretty clear on the idea that dumping untreated wastewater, specifically domestic sewage, which contains human fecal material, into lakes and rivers is a bad idea. And one of the main reasons that it's a bad idea is that there are many pathogens in human fecal material, and exposure to these pathogens in the receiving water can spread disease. But another reason we don't want to go around dumping untreated wastewater in lakes and rivers is because it will exert what's called an oxygen demand in that water body. So what that means is that bacteria living in the environment, and these are not necessarily pathogenic bacteria, will see the organic matter in the untreated wastewater and think of it as food. These bacteria consume this food and thrive and grow. And while they are growing, they need to respire, that is, use oxygen. If there's too much food, so too much sewage, then the bacteria can grow so well and so robustly that they consume all the oxygen in the water body. At that point, there are other bacteria that can step in, like the nitrate-reducing bacteria I mentioned before in the context of sewers. Bacteria will continue to grow in the absence of oxygen. But the point is that the oxygen is now depleted. This causes all sorts of problems for the aquatic life in that water body. Fish, for example, need a certain amount of oxygen to live. And low oxygen in a water body can set off a whole cascade of unwelcome ecological events. This is definitely something we want to avoid. By the way, this whole process of bacteria consuming waste as food and using oxygen in the process is exactly what we are trying to replicate in biological wastewater treatment. We grow good, happy bacteria in the presence of oxygen to degrade the organic matter in a controlled environment. And so that's what's shown here in this graphic. These bacteria use oxygen to grow on the organic matter, and in doing so produce more bacteria, plus water, carbon dioxide, and ammonium. Shown here is ammonia. And just to get into the details here, the genus Acetobacterium is thought to be responsible for most BOD degradation in activated sludge systems, along with some other genera. It's thought to be a pretty diverse mix of bacteria from different phyla in activated sludge. So probably also a diverse mix of bacteria out in the environment that can degrade wastewater. And just a reminder that we talked about activated sludge in part 3 of this series. So we want to avoid oxygen depletion in receiving waters. To do that, we want to know how much oxygen demand we are exerting on those receiving waters when we discharge wastewater. So we need to be able to measure the oxygen demand in waters, including effluent wastewater. The most common way we measure oxygen demand in the US is with BOD, but the most common way around the world is with COD. So I'm going to talk about both. Again, BOD is biochemical oxygen demand and COD is chemical oxygen demand. Not surprisingly, given their names, BOD is a bioassay while COD is a chemical assay. The BOD bioassay involves starting with a known amount of wastewater, spiking that wastewater with what are called seed microorganisms, and then storing the sample in the dark for 5 days at 20 degrees C. The dissolved oxygen in the sample is measured at the beginning of the analysis and after the 5-day incubation period, and the difference between the dissolved oxygen at the start and the end of incubation is used to calculate the BOD. Because the standard incubation period is 5 days, it takes 5 days to complete the test, and it is called BOD 5. COD, on the other hand, is based on a chemical reaction and the idea that organic matter should be fully oxidizable to carbon dioxide in the presence of a strong oxidizing agent under acidic conditions. So for COD analysis, the strong oxidizing agent is usually potassium dichromate, and the acidic conditions are generated through the addition of sulfuric acid. The amount of organic matter in the sample is back calculated then from the amount of oxidant required for the oxidation reaction. The COD test can be completed in 2 to 3 hours, but as you can imagine, COD is measuring oxidizable organic matter that may not be readily accessible to microorganisms as food. Therefore, COD values in raw influent tend to be higher than BOD values, usually about twice as high. So typical BOD measurements in normal strength municipal wastewater are in the range of 100 to 350 milligrams per liter, whereas COD will normally be in the range of 350 to 550 milligrams per liter. Again, that's untreated wastewater. And as we talked about in part 4, most discharge permits are written around BOD, not COD, in the US at least. And most require that no more than 30 milligrams per liter of BOD be discharged into the receiving water on an average monthly basis. So TSS, BOD, COD, nitrogen, and phosphorus species can all be thought of as conventional water quality parameters. And when we think about interpreting wastewater disease surveillance data, one of the things that's often done is to normalize the nucleic acid concentrations to account for really how much domestic sewage is in the wastewater sample. And way back in part 1 of this series, we talked about the different components of wastewater, and that the stuff that we flush down the toilets is only one of those components, and there are many others. So there are different ways to normalize gene copies per liter, but probably the most common is with the use of a fecal indicator using molecular methods to quantify the presence of that fecal indicator. So something like pepper mild monovirus or crassfodge. But there are other ways to normalize pathogen nucleic acid data in wastewater, and there has been a good amount of work looking at using the sorts of conventional water quality parameters I just talked about. So I thought it was worth mentioning the results of some of those studies here. And these are all for COVID or SARS-CoV-2, by the way. So Hutchison et al. Found that for influent samples from 2 wastewater treatment plants in Kansas, normalization with ammonium and BOD did not improve correlations with 21-day cumulative case counts for COVID. Lee et al. Found that for samples from 9 collection system sites in Michigan, normalization with TKN resulted in strong correlations with cumulative cases during the 10-month study period, whereas normalization with BOD or TSS did not. Malbarret et al. Found that for influent samples from 12 wastewater treatment plants in Alberta, Canada, normalization with ammonium, TKN, and TP improved correlations with 7-day average COVID case counts, whereas normalizations with BOD and TSS weakened the association. But the difference between normalized and unnormalized associations was not statistically significant. And Sweet Apple et al. Found that for samples from 394 sites in England, normalization with ammonium or phosphate, that's orthophosphate, improved correlation with 3 indicators of COVID prevalence, which is all to say that normalizing with BOD or TSS likely does not improve correlations between wastewater SARS-CoV-2 RNA concentrations and COVID case data, but normalizing with ammonium, TKN, phosphate, or TP may help improve correlations between wastewater SARS-CoV-2 RNA concentrations and COVID case data. But there's still some more work to be done there. I just wanted to cover one more class of methods in standard methods, and that is microbiological examination. Most of these are culture-based methods, such as for heterotrophic plate count, colophages, fecal enterococci, iron and sulfur bacteria, Legionella, Leptospira, and so on. But there are also staining and epifluorescence microscopy approaches for the direct total microbial counts, and bioassays analogous to BOD measurements. That's for the biodegradable organic matter. To my knowledge, none of these methods rely on molecular techniques, but this may change in the future. On here, you'll see coliform, fecal enterococci, and E. coli. These are all examples of fecal indicator bacteria, which are surrogates used to measure the potential presence of fecal material. And I wanted to talk just a little bit more about the 2 most common fecal indicator bacteria that show up in NPDES permit limits. And again, we talked about permits and the National Pollutant Discharge Elimination System in part 4 of this series. So the first is fecal coliform bacteria. So these bacteria are defined by their ability to ferment lactose. Very specifically, they are facultatively anaerobic, gram-negative, non-spore-forming, rod-shaped bacteria that ferment lactose to produce acid, gas, or both within 24 hours at 45.5 degrees C. There are many genera in the family Enterobacteriaceae that meets this definition, but most of these are not pathogenic. And the presence of fecal coliforms in water may suggest that the water has been impacted by fecal material from humans, so sewage, but it could also have come from animals. The most common type of fecal coliform is E. coli. So NPDES permits are increasingly being written around E. coli limits rather than fecal coliform limits. Not all E. coli, of course, is pathogenic, but it is a better indicator of recent human fecal contamination than fecal coliforms. E. coli and fecal coliforms are, of course, not the only fecal indicator bacteria out there. The larger circle I'm showing here includes total coliforms. So those are like fecal coliform, except that they thrive at lower temperatures than fecal coliform. So they can really come from anywhere, not just animal and human intestines, but also out in the environment. So they really aren't used anymore for indicating fecal contamination of a water. And there's also enterococci that are used more frequently as indicators for fecal contamination in recreational waters. But fecal coliforms and E. coli continue to be the most relevant for wastewater treatment facilities. To review the terminology, a grab sample is an aliquot of wastewater collected at a single point in time, whereas a composite sample is a wastewater sample comprised of blended, discrete aliquots taken at a regular interval over a specific period of time. And you are already likely quite familiar with this distinction. TSS is total suspended solids, which is the quantity of solids retained on a glass fiber filter and remaining after evaporation of water, usually expressed as milligrams per liter. TKN is total Kjeldahl nitrogen, which is a measure of both free ammonium and organic nitrogen compounds, usually expressed as milligrams of nitrogen per liter. TP is total phosphorus, which is a measure of all phosphorus compounds, including orthophosphate and organic phosphorus, usually expressed in milligrams of phosphorus per liter. BOD is biochemical oxygen demand, which is the measure of the biodegradable organic matter via bioassay, usually expressed in milligrams per liter of 5-day BOD. We often drop the 5 subscript from BOD and just assume that all BOD measurements are taken over 5 days. COD is chemical oxygen demand, which is a measure of the biodegradable organic matter via chemical oxidation, expressed in milligrams per liter of COD. And here are some questions to ask your utility partner related to their water quality monitoring that may help inform your wastewater surveillance program. The big overarching question is, what do you regularly test for in your raw influent, either for permit compliance or for process control? Some sub-questions related to that overall question include, which of these are measured on grab samples and which are measured on composite samples? How frequently do you analyze for each? Which type of glass fiber filter is used for your TSS measurements? And how easy is it for you to share your raw influent data with your health department and lab partners? And here are some relevant resources. If you want to do a deep dive into BOD, it's an assay that was developed starting back in the 1800s. I recommend you check out The Third Century of Biochemical Oxygen Demand. Interestingly, in that book, the authors note that, quote, many wastewater professionals have predicted the imminent demise of the test. They're talking about the BOD test. They go on to say, but it is still here and is likely to remain so for the foreseeable future, given how deeply it is embedded in the regulatory framework that surrounds the industry. Many of the methods used by wastewater treatment facilities to measure compounds to demonstrate permit compliance are in the Clean Water Act method summary. And finally, you may be interested in browsing standard methods itself. This was part 5 of Wastewater 101, How is Water Quality Monitored? Other parts in the series include part 1, What is wastewater? Part 2, How is wastewater collected? Part 3, How is wastewater treated? Part 4, Where does treated water go? And part 6, Who works in the wastewater sector? Please email nwbe@wef.org if you have any questions about this content or would like us to develop a custom training program for your organization. Thank you for tuning in.